Hey everyone, so today we will be dealing with gluconeogenesis. So, as the name suggests, gluconeogenesis, which means the formation of glucose from any non carbohydrate precursor. So, that non carbohydrate precursor basically, which we have to study, is of like four examples are given, which is lactate, second one is propionyl, coenzyme A, glycerol, and gluconeogenic amino acid. Glucogenic amino acid, glucogenic amino acids. So glucogenic amino acids will enter into the this pathway gluconeogenesis and lead to the formation of glucose. Okay. तो जैसे कि आप लोग को दिख रहा होगा कि ये जो pathway है basically similar लग रहा है glycolysis के, and the enzymes also involved in this pathway is similar to glycolysis, but this is not the reverse of glycolysis. Okay, it's not like that. And in glycolysis we saw that some steps were irreversible, okay, which was the first step, the third step, and the ninth step. So to circumvent these steps, suppose let's consider a precursor, a non-carbohydrate precursor, lactate enters into the gluconeogenic pathway through pyruvate only. Okay, so lactate has to form glucose, this glucose over here. Okay, so for that we have to circumvent these irreversible steps, which is the ninth, third, and the first step. For that we have some key enzymes of gluconeogenesis. So let's deal with it. Okay. So lactate is first converted into pyruvate, then pyruvate is converted into oxaloacetic acid, and this is first key enzyme, which is pyruvate carboxylase. So one carbon dioxide will come and pyruvate will be converted into oxaloacetic acid through pyruvate carboxylase. Then oxaloacetic acid will be converted into the phosphoenol pyruvate through PepCK. I'm sorry, wait. Hmm, PepCK, which means phosphoenol pyruvate carboxykinase. And 1 GT will be converted into 1 GDP. Okay. Then the reversible steps will occur through the same enzyme of glycolysis. Okay. And when the third step comes, to circumvent this step, we have fructose 1,6 bisphosphatase, which will convert this first fructose 1,6 bisphosphate into fructose 6 phosphate, and then we have the glucose 6 phosphatase to convert into glucose. So basically in exams we have to write the whole reaction with these four enzymes should be written. Okay, these four key enzymes of the gluconeogenesis is very important in PepsiChem. Okay, so one more thing, that this oxaloacetic form has to be transported into my cytosol, because the further reaction will occur into the in cytosol. Okay, so oxaloacetic is converted, transferred into cytosol through a shuttle, which is malate aspartate shuttle, which I will be dealing now. Okay. Hmm, hmm. Okay. So we have mitochondria over here, and we saw the formation of oxaloacetate. So oxaloacetate will form malate, and then malate will be transferred into cytoplasm. Okay, and this is malate dehydrogenase involved over here, which means like from oxaloacetic acid will be converted into malate by the utilization of one NADH. Then in cytoplasm again malate will be converted into oxaloacetate. Okay, and sometimes when we need uh, oxaloacetate from cytoplasms to mitochondria, then it will undergo transamination to form aspartate. That's why it's known as malate aspartate shuttle system. Okay, so aspartate will then enter into the mitochondria, and then it can be again converted into oxaloacetic acid. Okay, so this is malate aspartate shuttle system. Now, if we talk about the substrate, we have already talked about these four substrates which we have to learn. Okay, then regulation. So regulation, it's also very easy. We have three enzymes which we can control. Sorry, two enzymes which we can control. One is pyruvate carboxylase. This one. Mm, this pyruvate carb. Oh, sorry, pyruvate carboxylase. This one. So basically, this get activated by acetyl coenzyme A. How we can remember this? That if we have acetyl coenzyme A, which it will lead to the formation of citrates. And if citrate is increasing, which means citrate has to react with someone, and citrate reacts with, okay, oxaloacetic acid something. I mean oxaloacetic acid is converted into citric. Or we can just remember it like that. That acetyl coenzyme A is an activator of pyruvate carboxylase. Okay. Then we have fructose 1,6 bisphosphatase. Okay, this one. This this we have to remember that this is activated by citrate and inhibited by AMP. Okay, inhibited by AMP and fructose 2,6 bisphosphate. Okay, and ATP. As we know that gluconeogenesis, which means formation of glucose, requires energy. Okay, and glycolysis will produce ATP. So basically, if we have ATP, we can lead to the formation of glucose. So that's why ATP will enhance gluconeogenesis. Okay, then hormonal regulation. So as we know, insulin decreases our blood glucose level. So if we have to convert glucose into pyruvate, insulin will give a positive effect because its role is to decrease blood glucose level. So it will use the glucose and it will convert it into pyruvate. But if we need formation of glucose, then insulin will act as an inhibitor, and glucagon and glucocorticoids, which is cortisol. Cortisol, the function of cortisol is to increase the blood glucose level. So glucagon and glucocorticoids will increase the blood glucose level, thereby enhancing the gluconeogenesis. So this was about the regulation of gluconeogenesis, and now about the energy requirement. So three steps: pyruvate carboxylase, PepCK, and phosphoglycerate kinase. I'll show you this step. When pyruvate is converted into oxaloacetic acid, it will require 1 ATP. When oxaloacetic acid is converted into phosphoenol pyruvate, it will require a GTP, or equivalent is 1 ATP. Okay, and this step, this step over here. This was a reversible step. Still, here 1 ATP is utilized. Okay, so this step will require 1 ATP. But if this is single, then only half of glucose molecule will be formed. So we are taking 2 pyruvate. So 2 pyruvate will be converted into 2 oxaloacetic, and it will use 2 ATP, utilize 2 ATP. Then 2 oxaloacetic acid will be converted into 2 pep. I can show you over here. This step will like take up 2 ATP. This step will take 2 ATPs, and this will take 2 ATP. Okay, so basically it will utilize 6 ATP. Okay, in total. As a summary, we can say that if glucose is converted into lactate, as in case of glycolysis, then 2 ATP is generated. And if lactate, we took this, right, a non-carbohydrate precursor, lactate. Okay, if lactate is converted into glucose, it will utilize 6 ATP. It will use 6 ATP for the formation of glucose. So this was all about our gluconeogenesis. In that, for summarizing it, I can say first we need its definition, then we need to say about its site, which is basically cytoplasm or mitochondria of liver or renal cortex. Then we have to say about its substrates. Then we can tell about its four key enzymes. We can write the steps. We can write about malate, aspartate, shadow system. we can write about its regulation, its energy requirement, and at last we can write about this reaction. Okay, so this topic is an essay topic and it's kind of very important to understand. And initially, it's very complicated to understand sometimes because of involvement of so many enzymes. But I try to make it simple by using less enzymes in it. And these are the four key enzymes which we have to remember. So thank you. If you like the video, kindly like it, share, and subscribe.